As I start, my name is Aboka Eli. I'm a medical microbiologist, just a fancy way of me calling myself an expert. But yeah, so before I start, I have an exercise because today I'm a teacher. And my first exercise, can we just all stand up for a minute? I have a question for you guys. So, yes. How many of you believe you'll stay alive if I remove all the bacteria from your body? If you believe you'll remain alive, you can sit down. If I remove all the bacteria from your body. Okay, we have one casualty over here. Anyone else? Yes, if you feel you will remain alive. Oh, okay. Okay. For how long? Okay, fair point. So, I see people know their biology. So yeah, you can all sit down. So I have another question. Between microbes and, we know what microbes are. Small organisms, from the word micro. So between microbes and humans, which ones do you think came first? Microbes. Oh, okay. At least now I know we are together. Anybody believe there are good viruses out there? Just by a show of hands, I can see 1, 2, 3, 4, 5. Okay, fantastic. So yeah, what is microbiology? Microbiology is just basically the study of organisms that are not visible to the naked eye. What do I mean by a naked eye? Like these ones. Mine are naked. So the ones who have 4 eyes, they have added something. But then we need something extra to be able to see them. Okay, most people think when we say we are studying microbiology, we spend the day looking at a microscope. Okay? But that's not the case, and I'm going to try and debunk that. So you have different types of microbes. We have bacteria. I think they were mentioned by, was it group 8 or group 9? I'm not sure. Yes, the sharp boys and girls. So we have bacteria. An example of bacteria is something like Salmonella, which causes typhoid. We've heard about H. pylori. Yes, that's an example of a bacteria. We've heard about Mycobacterium. We've heard about that. Okay, we have people here who have studied. So we have wide number of bacteria. Another microbe are the viruses. We know about COVID. We know about influenza. Yes. Any other virus we know? HIV, Ebola. Anyone else? Flu, influenza. We know about rhinovirus. We know about it. Yeah. So apart from the viruses, we also have the fun guys. We know about fun guys. I call them fun guys. Fungi or fungi, depending on your first English teacher. So fungi are mushrooms. Anybody who loves eating mushrooms? Yes. So those are examples of fungi. They are responsible for the big mugs. We know yeast. We know about yeast? Yes. So yeast is responsible for your beer. Those of you who are taking beer. So that's a microbe at work. And then we have protozoa. Protozoa, an example of a protozoa is something like malaria and the mignot that we usually deworm every 6 months. When was the last time we dewormed? So please go and deworm, guys. Apart from malaria, we also have algae. Algae is found mostly in oceans as food for fish. So microbes are quite interesting because we think we run this planet, but it's actually these little guys that run the planet. Anywhere you can think of life existing, you will find a microbe, okay? So, like I was saying, we normally assume that microbiology is just looking under a microscope to understand this invisible world. But nowadays, that is not where microbiology ends. It ends in the computer. So we start by, first of all, isolating a bacteria out there, and then we end up in a computer analyzing this microbe. So while most of you will spend your lives running away from germs, as a microbiologist, I spend my time or my day hunting for these microbes, going down in sewage. Actually, my My happy day is going to chota sewage. I enjoy that, yes. So there's somebody who said, scientists, we have imaginary stories. We're not scammers. Anyways, so yeah, I spend my day in the looking around the sewer lines trying to find some microbes. And when I find a microbe, I get very excited. Unlike you, when you find that somebody is infected, you're like, oh my God, God help us. So me, I get excited. Okay? So biology generally has moved from just being a field of observation. Nowadays, it has a lot of computer science. So we have what we call computational biology. And under computational biology is where we can design life. We know about DNA. So DNA are the instructions for life. And using computational tools, we are able to design life. So I can come up with a new bacteria if I wanted to. Okay? So tonight's main characters are these two guys. We have the bacteria and bacteriophages. They kind of exist together in nature. One is the prey, the other is the predator. Anyone wants to give an educated guess which is the prey, which is the which is the prey, which is the predator here? Yes. Bacteriophages are the prey. Predator. Yes, yes. So bacteriophages are the predators and the bacteria are the prey. So just like in nature, we have let me say the game parks, we have lions and antelopes. So again, in the microbiological world, we have bacteria and the phages that hunt them, okay? And that is how, that's how life is designed. There always has to be something that is hunting you. Anybody who knows what hunts humans? Don't tell me a lion, though. Yes, mosquito. Any other thing that hunts humans? Yeah? Mamba. We know what mamba is. Crocodiles. So, you know, I find God to be a very cheeky architect because he decided to introduce diseases so that they can control our population. Do you agree with me? The guys who said I give lies, or scientists, we lie. Am I lying? So, yeah, that's how life has been engineered. There always has to be a balance so that one thing does not dominate everywhere. Okay? So I'll start with the bacteria. So we have the good bacteria. Yogurt is an example of a product that is made from good bacteria. Okay? The lactobacillus that somebody mentioned. We have a type of bacteria that's called lactobacillus. Okay? Mala, maziwalala. That's an example of something that is made by the good bacteria. Okay? For the bad, I think these are the diseases now. But also for the bad, we have some that cause losses, not just the diseases. Because when you think of bacteria, most people will think they just cause disease to us humans, then you get sick. But if you're a farmer, like me, I'm a farmer, I'm a child of a farmer. So I was keeping chicken at home. And then I had 100 chicks, and I lost 30 of them in 1 week. Can you imagine? That was a big loss. So yeah, that is some of the bad bacteria. On the good side, we have actually some bacteria are able to kill other bacteria. For example, there's somebody who mentioned we have the, let me call it the microbe of the night, lactobacillus, which is found within the female vagina. So yeah, they exist there. And a common characteristic, if you want to know that these guys are healthy, the panty can sometimes get discolored. Okay? Yes, it is healthy. Okay? Sawa sawa? So in case you did not know, now you know. That's what causes that color change. Sometimes when you see there's some bad odor over there, you can tell something is not right somewhere. Okay? So I hope now you know. So on to the star of the night. And I'm wearing this very amazing brand over here. These amazing guys, what we call the good viruses. They're called bacteriophages. Discovered over 100 years ago. And they are the most abundant biological entities on the planet. So they are more than humans, they are more than bacteria, they are more than anything you can imagine on this planet. That number over there has I don't know how many zeros, but it's a nonillion. If you know how many zeros are those. But there are very many. That's all I can say. And they are very tiny, so you can't see them with the naked eye. It's an estimation, okay? Okay, let me give you a scenario. So, and I'm going to explain this shortly. For us to understand why these guys are good, we need to understand how a virus works. Like I said, they are good viruses, okay? So viruses are very specific. They have what we call a lock and key scenario, type of way of how they work. So a virus will have a key that only opens a certain lock on a particular cell, or in this case, a building block of life. So if it's a human cell, for example, in HIV, the bacteria, the key on the HIV will only unlock the T cell. T cell is one of the cells in your body that helps fight off diseases, okay? If it's COVID-19, it will have a key that is specific to cells within your respiratory tract. So it will not affect anything else aside from the respiratory tract. So up to there, we are together, right? So for the bacteriophage now, the key that it has is very specific to a certain type of bacteria. Okay, so let's assume all of us here are bacteria. Anybody wants to pick a name? Salmonella. Who is Salmonella? Yes, we have Salmonella. The guy at the back, yes, so you have Salmonella and Bacillus. He is Klebsiella, okay? So if I'm the phage, I have a key that is specific to Salmonella. That key will not work for Bacillus, it will not work for Klebsiella, okay? Sawa sawa? So up to there we are together. That's the specificity of viruses. That is how viruses will work. They'll only infect that which the receptors code for, okay? So, and a fun fact is viruses are not alive. Okay, they're not alive like us. Because for something to be alive or a living thing, it has to be able to grow, it has to be able to reproduce. Anything else? These are things you learned in biology. To die, yes. So a phage on its own, for example, if I was the phage, I'll just be here hanging, yes. Yes, a fetus is alive. It usually even has a heartbeat. Okay? So the phage or any virus, if it's here in the air, it will just be floating around. It can't do anything. Until at such a point it gets inside a living cell, is when now it can replicate. Okay? Yeah. So phages were discovered by this amazing gentleman. We have Frederick Twort in 1915 and Felix de Herel in 1917. Okay, and they discovered them independently. So everybody was doing their own thing, then they see something, and they're like, there could be something here that is killing a bacteria. Okay? Felix de Herel is the one who came up with the name bacteriophage, to mean bacteria eater. So phage comes from the Greek word phagein, to mean to eat. Okay? So that's why we have the name bacteria eater. Okay? He's also the first person to perform the first phage therapy. When I say therapy, therapy is just treatment. Okay? So it's just a fancy way of saying treatment, phage treatment. So what he did is he gave himself an infection. I think it was a dysentery. And then he ingested some phages and then the infection was cleared. That's how crazy scientists were back then. Yeah. They would do experiments on themselves. And they would steal dead bodies. Okay? So this is how phages work. Somebody was asking, how does it get inside the cell if it's not alive? So when the phage is floating around in the environment, okay, it has the tail or the things you see as tails at the end, they are the keys. We call them receptors. So they will attach to a bacterial cell. So it will just be floating around, then it finds a cell, it attaches to it if it has the specific locks, okay? Then once it attaches, it will release its genetic material or its DNA its instructions for life into the cell. Okay? Once it gets inside the cell, they hijack the cellular machinery of this bacteria. I hope you understand when I say cellular machinery. Or so in this case, think of the bacteria as a factory. When the phage comes in, or when it introduces its genetic material inside, it starts creating copies of itself. Okay? Then the copies of itself will keep on generating. So how it creates copies of itself, it will generate, it's like, assume me I'm a phage, so it will generate an arm, it will generate a leg, the head, like that, and then all these things will come together. Once they've assembled, they will burst up the bacteria. So the bacteria will die, and then the cycle will continue. So the particles that are released will go and attach to other bacteria, and the cycle will keep going until our bacterial population in this environment is completely cleared. Okay? So that's how phages work. If this is happening within your body has a way of clearing anything it does not want. Okay? Again, God, the amazing architect of life. So this is how we do a phage hunt. As you can see, I call myself a phage hunter. So yeah, this is how we do the phage hunt. We go to the field. I've told you guys that I spend my day, what is kuchokora in English? Foraging, yes, let me use foraging. I spend my day foraging sewage. Okay, so I collect a lot of sewage. Actually, this day we were with my supervisor. I was the one taking the picture, of course, yes. That's why I'm not in the picture. And people were passing around asking us, why are we collecting stool? So we were collecting sewage from a slum somewhere. Once we collect that, remember I said phages are very tiny, okay? So we'll bring that sample to the lab, and then we'll process it. How do we process it? We filter this sample. We filter the sample using very tiny filters. So kichungi kama ile ya chai, but sasa very tiny, okay? We use the 0.22 nanometers. I don't know if you know how small a nanometer is. It's very, very tiny, okay? So that what we expect is in our filtrate, we will have removed everything else from this sample. Any debris, any poop that was there, any bacterial cells, so that in our sample, we only remain with the virus particles, okay? Then that virus particle, we get to grow our bacteria on a plate. Yes, we do give our bacteria food. So we put food on the plate, then we grow our bacteria there, uniformly across the plate, as you can see that plate. I don't know if you can see, but at the middle, there's like a clearing, if you can see that clearing. So when we see that clearing, remember I said biology started by just observing. So that clearing is our phage killing the bacteria that we had grown on the plate. Okay? So that's how we do the hunt. So we'll keep doing the rounds of purifying this phage until we have a phage formulation that can now be used for treatment. Unfortunately, for Kenya, we are not here yet at the formulation stage. We're just isolating them and trying to study them and see how they work. Okay? And we do this work with this amazing organization. It's a non-profit. You can look us up on LinkedIn if you want to follow our work. And we do just do the work of training young scientists to go out to the field and hunt these bacteriophages. We're also doing some advocacy work, like the work I'm doing here today, so that the next time you're in a hospital, you can tell the doctor, have you heard about phage? Yeah, so you can brag that you know science. Yeah? So it's part of advocacy so that when these little guys get to market, you don't get scared saying, I don't want a virus getting into my body. Okay? So these are the organizations that do phage research in Kenya. We have ILRI. ILRI have done some amazing work with chicken. They isolated phages against salmonella. So salmonella, as much as it affects human causing typhoid, it also affects chicken. And it can cross over from chicken to you. Okay? So quite interesting. So they've done some studies to use phages to try and reduce the contamination levels of salmonella in chicken. We have us there, Phage Hunters Training and Research Program, KEMRI as well, Jomo Kenyatta University. We have Kenya Institute of Primate Research and the Technical University of Kenya. So these are those that I know of. Others are doing it, but probably I haven't interacted with them yet. So phages have a lot of applications. In the medical side, they can be used to treat drug-resistant bacteria. Okay? Somebody mentioned what antibiotics are. We remember? What are antibiotics? Yeah? Yes, so takers of life, anti-life, in short. So when antibiotics don't work, we need something that can treat this infection, right? So phages are a known alternative, as you have seen from Felix de Herel, who treated himself, so they can work, okay? Then we have agriculture. So phages are organic. You have them in your stomach, they exist in nature, they exist in sewage, yes. So they are organic. Yeah, sounds crazy. Yeah, so how we make them get into the body is we do a series of purifications. We have to sterilize them. We can't just take them from sewage and inject them in your body. Come on. Yeah, now I'm saying it. Thank you for asking. So in agriculture, for example, if I wanted to produce organic chicken, and normally most farmers will use antibiotics in livestock production so that you don't incur losses. So phages, being they are very specific, they won't kill everything and they won't remain. For example, trust me, the chicken you eat, maybe from KFC or the one you eat at home when you go to Shago, was treated with antibiotics, and most antibiotics usually have the withdrawal period, ideally 14 days. So if you were administering antibiotics, you need to wait for 14 days before this chicken can be consumed. But then if I'm a farmer and then I was giving this chicken antibiotics, and it's almost, somebody comes today, like I gave them antibiotics in the morning, and then somebody comes and they tell me, I have 1,000 here, I need chicken. You think I will wait for the 14 days? With this economy? So that's what's happening. So that's why we're trying to advocate for phages in the agricultural space to just reduce the misuse of antibiotics there, because they are highly unregulated. I can quickly go to any vet and buy any antibiotic I want to use in my farm, okay? Then we have in food safety. In food safety, for example, the mutura somebody was talking about. When you go and buy mutura, imagine if that guy had some phages and they're spraying on it so that you don't get the salmonella, okay? So we can use them for food safety. And they are organic, I will stress that again. Biotech and diagnostics, and this is the fun part, and I'll explain why it's fun. Just like your body, when something is invading, your body will fight it off, right? So the bacteria will do the same thing. They will also try and fight off these invading viruses. So what they use to fight off these viruses, it's called CRISPR-Cas9. Please, just say CRISPR-Cas9. I know I'm now things are getting deep, eh? CRISPR-Cas9. So how CRISPR-Cas9 works is when the phage comes in, it will inject its genetic material, right? Then these genes will go and incorporate with the bacterial genes. We are together up to that point. So at this point, the phage genome is within the bacterial genome. So the CRISPR-Cas9 will do like a check of this bacterial genome. So it will like start going around it and see, oh, this is something that is not supposed to be here. And then it cuts it out. Okay? So that's how bacteria will protect themselves. Now, scientists, the imaginary people, we were like, how can we use this? So using the same technology is what we are using to edit DNA. We've heard about gene editing. Yes, so that's now the technology for gene editing. Okay? So it was observed in a bacteria how it's preventing it from Nini, and then now we are using it to try and exploit further, okay? And it's a whole conversation. I won't get into this. An industrial application. For example, let's say we were making beer using yeast. And then, God forbid, some bacteria got inside there. Bacteria that is not supposed to be there. Then it starts producing other, we call them metabolites, let me call them chemicals, that can cause the taste of the beer to be bad. So how do we prevent this? We can introduce phages that target these bacteria so that these bacteria will not be able to grow within the beer. That's just one example. And there are very many other examples which I won't get into. You can go read after class. So as I was saying, for the biotech is where things get interesting. So according to Forbes, phages have been dubbed the next $1 trillion idea in biotech. Yeah, I'm looking forward to being the next trillionaire after Elon Musk. So, and it's a whole field that's just coming up. No Elon Musk. Okay, I'll be the first Aboka trillionaire. We are together. So it's a whole conversation that's happening, and that's why everybody is buzzing about phages, because they're trying to, how do we get a share of that $1 trillion cake? Okay? So now, which brings me back to antibiotics, which are humanity's greatest medical discovery. Why are they? They were discovered in 1928 by a guy called Alexander Fleming. So how he discovered them is a very interesting story. So this guy was growing some bacteria in the lab. So he was a microbiologist like me, growing some bacteria in the lab. Then he decides to go for a holiday, about 2 weeks. So when he comes back, he gets a contamination on the bacterial plate. So this contamination was a mold, like the one that grows on bread. Okay, so this mold, I've forgotten the name. It's a very complicated name, so I won't get into it. Around this mold, bacteria were not growing. Okay, so he was like, probably this thing is able to kill this bacteria, and that is how penicillin was discovered. Okay, so before penicillin, we had what we call the pre-antibiotic era. So this was before antibiotics. How did people used to treat antibiotics? Do you have GOT fans in the house? Winter is coming. Yeah? Yes. Yes. So, before antibiotics, how people would treat. So, there's a guy in GOT, spoilers ahead, who gets his hand chopped off. And how they treat him is they put his hand, or the remaining part now, in boiling wine. So that was how people used to treat wounds back then. I'm sure you've seen this in movies whereby somebody will burn a hot, like a blade, and then put on the wound. Yes. So that was how we used to treat infections. Other crazy ideas was people would pour even sulfuric acid on the wound. It was that bad. Yeah? So then we have the antibiotic revolution. And in the 1940s with World War II, people were having infections, etc., etc. Penicillin was there, it was easy to produce. We started producing it in mass. So these are some of the examples of antibiotics. I won't go into them because I'm sure they were mentioned. We have amoxicillin. Azithromycin is one I'm sure you guys use a lot. Misuse is the right word. So in the 1940s, there was like a boom of antibiotics, the wonder drug. Penicillin is here. And there's this famous poster that was going around during the war times that thanks to penicillin, he will come back home. So now people are not dying from wounds as they were dying before. There's also this amazing one whereby people are using also again penicillin to fundraise. This was just where they're saying if we finish this building faster, the quicker our wounded men get penicillin. So they were fundraising for a building to produce the penicillin. Okay? I love this one. Yes, can treat gonorrhea in 4 hours. So that was what penicillin was. It was a wonder drug. It could treat anything and everything. Do we know the symptoms of gonorrhea, by the way? Hello, wanaume? Hello? Men? Hello? Hey, you're not talking, men. So, when you have gonorrhea, a common characteristic is you have swollen gonads. So you walk around like this. Yeah, so it's a very, so HIV is not the only STI, guys. Please stay safe. So, and this is why antibiotics are important, because most of modern medicine is a success because of the success of antibiotics. So when they don't work, we are in deep trouble. For example, you're able to go to hospital and get a successful surgery because antibiotics will treat you, will treat the wound. But imagine a case you go to hospital, you have surgery, and then after you leave surgery, you get a drug-resistant infection that you not even work with in the hospital, but you found it there in the hospital, and then now it's not even responding to the drugs. So what do? We start changaing pesa ya matanga. So a lot of children in the neonatal unit as well are also dying due to antibiotics failing. So it's a real problem. Okay? So I'd just like to focus on this, what happened in the 1940s with so somebody would ask, if phages were discovered in 1915, 1917, antibiotics came later. Why is it that antibiotics are commonly known and phages are not known? You know, good question. So reason being, there's a lot of politics that affect science. So when phage was discovered, Felix de Herel did a lot of work with the former Soviet Union, Hitler. Yes, you know the stories of Hitler and what he did, the crazy experiments. So the science they did had a bad rep. Regardless of even if you were doing great things, it was bad. So most of scientists during that time, they chose to adopt what the West were doing. And the West had really adopted antibiotics. As you saw from the previous posters, they were really pushing antibiotics. So they were adopted quickly. Number two was because antibiotics were easy to produce and we could understand them. An antibiotic, for example, penicillin, we had Salmonella, Bacillus, and Klebsiella. So penicillin would kill all of them, okay? But phage would only work on, let's say, if it's a phage targeting Bacillus, it will only target Bacillus. It won't go for Klebsiella, it won't go for Salmonella. So it depends with the particular type of phage. So we can have a phage, how do I say this in a simple way? So E. coli is a bacteria, okay? And it has cousins, brothers and sisters. So we have like enterogenic E. coli, pathogenic E. coli. So call them like brothers and sisters of E. coli, okay? So we can have a phage that will attack 6 sisters and miss one brother. And then we'll have one that will only attack the brothers and miss this one. So it's a game of chance. And I'll explain how we apply phages in therapy. So whenever we want to apply phages in therapy, we use a phage cocktail. People who love cocktails, hello? Yes. So a cocktail is just a mixture of phages. So we take different phages targeting E. coli, and then we put them together so you have an E. coli infection. Then we administer that infection, okay? So here what happened was, in the West, antibiotics were easy to produce. We could produce them in mass, in large quantities. Phages, on the other hand, in the early 1900s, the field of virology had not even started. So what these guys were saying is, within this liquid, there's something that can kill a bacteria, and we don't know what it is. And if they administer it to, let's say, a patient having typhoid, this was a phage targeting Salmonella, it will kill it. But then you try it on another patient, it does not work. So the results were not consistent. And that's why antibiotics really gained traction and phages strolled. However, in Eastern Europe, the institute, Eliava Institute, kept on doing the phage research. Up to date, they still produce phage cocktails that are used to treat patients. And I'll be mentioning some cases of patients that they treated. So when Alexander Fleming was receiving his Nobel Prize in 1945, he gave a warning about antibiotics. So he said, a time may come when penicillin can be easily bought from the shops. And the ignorant man. Are we ignorant? Yes. So the ignorant man and woman will go and buy this penicillin and expose non-lethal quantities or underdose himself. And by so doing, will expose the microbes to non-lethal quantities, making them resistant, because he could see it in the lab. What I mean is, you have an infection, but then you dilute your drug. Let me say dilute your drug. So it doesn't kill everything entirely. It leaves something. And in the lab, he could already see some of these bacteria were becoming resistant, okay? So we didn't listen, unfortunately, and that's why we are here. We call them superbugs. So you will start with a population like that in yellow, and then natural selection is just a fancy way of saying survival for the fittest, okay? So you have some people who have gone to the gym. Let me call the bacteria that have gone to the gym, and they have some good muscles. They can fight off antibiotics, okay? So when you introduce the antibiotic, it will kill everything else, and then it will leave some of the fit bacteria. So these fit bacteria will start multiplying, and they will be the majority within this population. So the next time you introduce an antibiotic, it won't work, okay? And these bacteria usually have, let me say they mutate. They have different mechanisms around how they fight off antibiotics. Some of them, for the antibiotic to work, it has to get within the bacteria so that it can work. But this bacteria will take in the antibiotic and then pump it back out. Okay? Some of them will produce enzymes that are able to break down these antibiotics. So the antibiotic that was supposed to work, it will remove the thing that kills it from that antibiotic. Okay? Some of them, you remember the keys and locks I was talking about? So also, the antibiotics, sometimes they usually attach to those receptors. So a bacteria can decide to change its skin so that the next time this antibiotic comes, it's not able to recognize the bacteria, and then it survives. Some of them will even grow a thick skin around them and prevent themselves. So they have different ways of maneuvering the antibiotics. And this problem is projected to cause, we'll be losing 10 million people by 2050 every year. So you can try imagine how many people those are. At the moment, you're losing about 1.4 million yearly. Okay? So we have one problem, the antibiotic resistance. And we have two strategies. And what I want you guys to pick from this lecture is bacteriophages are not going to replace antibiotics. Okay? So it's just an additional tool within our toolbox to fight off antibiotics. Okay? So not antibiotics, bacteria. So antibiotics will work as a nuclear bomb. They will kill everything. They don't care. Okay? For the bacteriophages, it's like a sniper. It's very specific. So it will only kill the specific person that it has been sent to kill. It will not care about the good bacteria. So if you've been taking some probiotic yogurt, it will not care about the good bacteria that you've been drinking. It will only go to that which that is causing infection. Okay? And that's one of the major advantages we have against antibiotics. So like this, the image on your left, the bacteriophages, this is a product produced by Eliava Institute. So if you're in Georgia, you can just go to the shop, maybe you have a stomachache and you buy some Phages and you pop the drink, okay? Instead of buying antibiotics. So when antibiotics fail, imagine this here is a lab report of somebody who has a bacteria that is resistant to everything that is being administered. I don't know if you can see the image, but the image on the right shows those are different antibiotics that have been tested, and all of them have come out resistant. So imagine you're in ICU, and the drugs that are supposed to save your life are not working. What do? And your doctor does not know about Phage. So that's part of the reality of where we are at. So these are examples of people who had such a scenario. We have Tom Patterson. I love their stories because they've influenced my career in a big way. I had Tom Patterson's story back in 2019 when I was still doing my undergraduate. And it was a TED Talk how her husband, how sewage saved her husband's life. So luckily for Tom Patterson, he had a scientist. The wife was a scientist. So watu mlikuwa mnasema wanasayansi, we are imaginary. Please get yourself a scientist. Okay, so it's a very interesting story how she interacted with scientists from across the globe, and they got together, made a phage cocktail that his husband was in a coma, and after 3 days of being administered, he got out of a coma. Okay? Christopher Schaffer's story is also another fun story. So for him, he saw when he got the infection in the hospital. So he could see how the doctors were handling, he was going for surgery, and he saw how the doctors were handling the tools for surgery. They were not aseptic. So in his mind, at the time, he didn't know about antibiotic resistance. So he contracts a superbug, it was E. coli, that was responding to no antibiotics. And then he had to travel all the way from the US to Georgia so that he could get that treatment of phage. And this was after 2 years of continuous antibiotic therapy that was not working. So he was literally almost dying until he went to Eliava. And 4 months down the line, after starting the treatment, he's alive and well. If you go on our page, you'll find his story. Okay? So when can a virus save a life? there are some key things that need to be met for a virus to save your life. Number one, you need to have a severe illness. So in that, what I mean by severe illness, you're dying. Yes, so it needs to be resistant, what we call the superbugs. And then there need to be no other alternative. So the resistance bit, no other alternative should be able to clear the infection. Okay? And then now there should be no other trials, so that is no current drugs that are under trial to try and solve the same problem. So that now we can say, oh, let us try phage because it is safe. Okay? So it's also, its benefits need to outweigh the side effects of this new drug. So how do you request for phage therapy? Number one, you need to talk to your doctor. So your administering physician, you need to convince them that, yeah, you've told me that I have this infection that is not responding to antibiotics. Can we try phage therapy? So now that you know about phage therapy, the next time you're in hospital, ask the doctor, have you done antibiotics test to confirm that the antibiotic you're giving me will work? Okay? Then you need to contact somebody who makes these phages. We have different companies across the globe, including the Liava Institute, and then you need to get approvals from the right regulators, including ethics guys, for you to be able to use these phages. This was mentioned before, and it's just me stressing it. Antibiotics remain the cornerstone of modern medicine. We can't do without them. So how do we make sure that they remain working? Only use them when they are prescribed, so don't self-medicate. Complete the full dose. Don't leave them so that you share with somebody else. And then stay clean, guys. I know I work with sewage, but I wear gloves. Okay? So you guys don't wear gloves, so make sure you're walking around with some sanitizer. When you go to the toilet, don't just leave and come and eat the fry. My God. Okay? Sometimes. Yes, that's a very good one. So the phones are a very big source of contamination, by the way. Okay, so don't go with them to the toilet. If you go with them, make sure as you leave, you sterilize your phone. Sawa sawa. So, yeah, antibiotics don't work for colds and flus. I hope you know this. So how do you differentiate between a cold and something that is, and strep throat? Do you know what strep throat is? Not sore throat, but strep throat. Okay, so let me explain. So we have some throat infections that are caused by bacteria. Okay? So if you have strep throat is caused by a bacteria called Streptococcus pneumo something. I am trying to avoid those Latin names. So how such an infection would look like, if you look at the image this side, the tonsils will be swollen and will have some white patches on it. So that tells you that this is a bacterial infection. Okay? For the common cold and flu, common cold is caused by rhinovirus. It will just be inflamed. It will be red. There'll be no white patches. So if you have the red one, the cold, just take some hot water, eat some tangawizi, you will be fine. Sawa sawa. Don't go and self-medicate, please, guys. I repeat. So the next time you see somebody asking for an antibiotic and they have a cold, what do? You slap the hell out of them, okay? And tell them that's a virus, okay? So fun reads for the bookworms in the house. We have this one by Professor Strathdy, how she saved her husband using sewage. She coined that name the perfect predator. I hope now you know the perfect predator. You can go find that. It's an amazing story. I heard very soon it will be a movie, so I'm looking forward to that. Then we have The Good Virus by Tom Ireland, which is a book on the history. So if you want to read about how people used to steal dead bodies so that they would isolate phages from them and use them to treat people, that was where science came from, by the way. Yes, we used to steal dead bodies back in the day. And then finally is The Finding Phage, how Christopher Schaffer saved his own life using a phage. Okay? So brace yourselves, guys. Winter is coming, or it is here. Yes. So antibiotic resistance is coming. Please take care of antibiotics. Use them responsibly. And thank you. That is the end of my lecture.